Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Kalina from Business Review, and I will be your host today. It is our pleasure to have Encardia with us today, who will be presenting this webinar titled The Generation of hiPSC Models for CNS-Relevant Cell-Based Assays. Today's guest speakers are Stephen Dublin, PhD, Head of Product Management, and Isabel Anoffer, PhD, Cell Development Scientist. I would like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you do disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask any questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We will allocate some time at the end of the session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you in order to get a better view of the slides. But for now, please allow me to welcome Stephen Dublin. Stephen, over to you now.
Great, thanks. Yeah, thanks for the introduction, and many thanks to everyone for taking the time to join this webinar today. My name is Stephen Dublin. I work in product management at Encardia, helping to try and match clients' needs with our future innovations. In this short intro, I'll give you a brief overview of Encardia and how we support therapeutic development in drug discovery, and also increasingly in cell and gene therapy with customized iPSC solutions. And then my colleague Isabel will detail recent work at Encardia and the development and optimization of CNS models for electrophysiological assays, for compound responses and physiological co-cultures. That's the agenda. So about Encardia. So Encardia's mission is to bring better therapies to patients faster. And what we talk about, and we truly believe that it's this easier integration of stem cell technology into drug discovery and cell therapy development that can really make this a reality. So we've established over the past 10 years an integrated iPSC platform on which we provide a range of services and products to assist clients really in this process of efficiently implementing pluripotent stem cell technology in their development processes. And at its core, there are really three core elements to the Encardia platform, which is iPSC sourcing or reprogramming, directed differentiation to the desired cell type, with manufacturer always looking to scale, and assay of the cell model for identity and for functionality. So we're a globally operating company with client base that's split roughly between 50-50 between North America and Europe. Our headquarters is in the Netherlands in a biotech center with situated near leading biopharmaceutical companies and university medical centers. This location also houses our discovery technology and our manufacturing technology teams. And then it also serves as a secondary manufacturing site to our primary manufacturing operations and logistics facility, which is situated in Belgium. So the site in Belgium is ISO 9001 certified, and it's located with easy access to two airports so that we can reach international customers quickly and efficiently. We have business development professionals that are located in North America, also in Europe, and we cover APAC and the Middle East regularly as well. We also have a storage and logistics center located in the US through which we can quickly serve clients on both coasts and in between. So we operate across three core application areas, those being cell therapy, efficacy testing, drug discovery, and safety testing, of which we increasingly see safety as a component of efficacy and cell therapy projects. So for drug discovery clients, we generate custom human disease models for applications in phenotypic screens, usually trying to orient towards higher throughput, either at the client side or through Encardia's labs and partnerships. For cell therapy developers, we really focus on building robust manufacturing processes for cell models that scale to the required quantities of cells for their downstream therapeutic application, and also for preclinical investigation. So we see demands for larger quantities of cells for like animal studies and regenerative medicine. Through these activities, we've really generated significant experience in the area of really porting processes from early research studies to protocols really ready to scale and transfer into that technology, then either into the client's facility or to the client's GMP partners. Finally, we work in the area of safety, clearly. We have a variety of cardiovascular and CNS models for this purpose, and a menu of services to support safety assessment. We've been providing qualified cardiovascular cell models for use in safety assessment to pharmaceutical companies since pretty much the formation of Nkardia. And I really believe that this deep history in providing the commercial products gives us a really unique appreciation as a partner for our clients' pressures, not just to really conduct high-level research, but also to then, in the end, always be looking at delivering a commercial-ready product reproducibly. So, although I said that we work in these three distinct areas, we do see overlaps in the required technologies and skill sets, which we then leverage in our iPSC workflows for clients. So, this illustration represents how we really see the implementation of stem cell technologies and the discovery of various new therapeutics, so be they small molecules or cell and gene therapies. So a typical project with us starts with iPSC generation from healthy or patient material, then moves on to genetic modification or modulation, either repairing a mutation or, for instance, introducing a mutation or a reporter gene, then differentiating to the desired cell type, and then manufacturing that to the appropriate scale. For drug discovery and gene therapy clients, the cell is frequently viewed as a critical material in the service of testing for efficacy and for safety assessment. So we focus on developing assays to measure compound effects on the cells, often with the goal of implementing those screens in high throughput formats. For cell therapy clients, moving over to the right side, the cell is commonly the desired end product. So you want to make sure that the identity and functionality regarding efficacy is really clearly established throughout the process. And so we see that a lot of the technologies that we utilize and the assays that we're developing in drug discovery are really, in many ways, directly applicable and portable over to our cell therapy projects. In the end, for both drug discovery and cell therapy, the benefit of iPSC technology is this renewable cell source at scale that recapitulates this human physiology together with the qualified informative cellular assays. So for manufacturing of differentiated cells, we've developed protocols, and we continue to develop protocols for differentiation of iPSC cells to a variety of cell types and cardiovascular models like ventricular cardiomyocytes, which we execute in stirred tank bioreactors up to scales of 20 billion per run. We also have protocols for atrial cardiomyocytes, endothelial cells, smooth muscle cells, and we have protocols for astrocytes, CNS neurons, peripheral and cortical neurons, several of which my colleague Isabel will discuss shortly. So we routinely run these protocols at Nkardia and make those models available to clients. We also use them in internal services as well. So by manufacturing and applying the cells in these services, we really gain a deeper understanding of the biology and the application of the models and the parameters of the assays, which we can then directly transfer to our clients. And I should emphasize here that these protocols can be utilized with a client's iPSC line, or we can source a desired iPSC line for you and then perform that manufacturing using our protocol, which makes it a pretty convenient service. There we go. So it can be that there's a model that you're seeking that isn't commercially available. or the existing models really aren't matching your requirements for your process. And we can work with you to manufacture an appropriate model and even qualify that model for your functional assay requirements. We like to keep this relatively straightforward because, like I said, with the other ones, the cell model really is your material getting you to your screens. So what we do is, simply it's about defining what success looks like, and then developing that agreed requirements at the beginning. We'll go back and do literature research. We develop a scientific plan and assess the feasibility and risks associated with that project, and then establish a protocol. We'll do things like defining the culture system when required, characterization assay development, functional assays fit for your purpose if desired, and then proceeding to manufacture. We can optionally proceed to large-scale manufacture and then tech transfer that assay to your facility or perform it for you at Ncardia. So there are many ways to arrive at a differentiation and manufacturing protocol, clearly, but we really aim to build these robust processes that keep cost and efficiency in mind. So what you're seeing here on this slide is an in-house process for a cardiomyocyte model. On the y-axis, you see manufacturing costs for generating 1 million cells. And so for this process, we were able to achieve a 16-fold reduction in cost per million cells, along with a 50-fold increase in yield by optimizing the manufacturing protocol. So the key drivers of this process improvement were achieved by a reduction of costly reagents, so taking a look at what we were spending on materials like coating materials and looking at things like replacing growth factors with alternatives, eliminating extra handling steps, and also porting that, the biggest jump you see is really porting the existing protocol into controlled bioreactors. So we're very flexible in our process development. We're always looking at efficiencies. We're not platform dependent. We're very independent on how we build your process because we're building it to your specification. So, in general, each process has parameters that can be optimized, be it media and supplement, like we said, manufacturing formats, and even implementation of automation where necessary. And we do recognize that for specifically cell therapy companies, the concept of lowering the COGS is really something that's talked about a lot. But there's also an appreciation, I think, in the drug discovery area as well, because what these lower costs in manufacturing translated into is lower costs per test, and that you're paying to perform these drug discovery services. So with respect to with respect to the drug discovery and development using iPSC technology, we focus on cardiovascular and CNS diseases. So we're well equipped to perform drug discovery services from cell model generation to assays. I've already discussed the in-house protocols for model generation. We tie that together with our disease modeling capabilities, such as editing technologies, CRISPR, together with our partner Horizon, to introduce or repair mutations. We have projects that implement chemical induction of disease phenotypes and physiological co-cultures to more closely mimic this native biology, which my colleagues will discuss shortly. We have a wider range of assays and technologies for cell characterization and drug testing. This includes gene expression analysis, assessment of electrophysiological properties, morphology assessment, contractility, calcium signaling analysis, the plate reader-based assays, so for biomarker analysis like HCRF, alpha-LISA, metabolic activity assessment, and more. So you can see with the cell model generation and editing, plus the in-house assay development capabilities, we're able to perform projects spanning from target discovery and validation to preclinical development. So typically our target discovery projects that we're involved in involve validation of targets through phenotype rescue by proof of principle compounds. In primary and secondary screens, it's typically phenotypic screening, trying to find a molecule that reverses the disease phenotype. using a physiologically relevant cell-based disease model. Hit to lead and lead optimization, we're typically trying to provide insights to help establish SAR and quantify potency as well as identity, safety, liability of lead compounds. And for clinical, preclinical development, we have projects where we're introducing different genetic backgrounds into the test system to identify backgrounds in which the molecule is most effective. So starting a cell model generation screening project with us is relatively straightforward. So what I'd like to point out here is that we schedule a call with scientific domain experts who work with you to develop the specification for your project. Then what we'll do is we'll go back and develop a proposal that includes the definition of prices and deadlines and deliverables for your review and approval. Up to that point, really, your commitment has been the time. But then once that project is approved, then we get to work executing on the agreed plan. So I should say here, we're open to a wide range of business models. So we do basic service structures, fee for service, up to co-development and partnerships, looking at building assets. So there'll be contact info at the end of this presentation, and you can reach out to us directly through our website or your local business development partner to schedule a consultation. So with that, I'll hand over the presentation to my colleague, and thank you for your attention.
Hi everyone. Thank you all for joining us on this webinar. So my name is Isabella Noff, and I'm a cell development scientist at Encardia, where I work with our neuro portfolio, developing neuronal models and characterization and functional assays. And today I will be talking about our cell models for seizure and also neuroinflammation. But before that, I would just like to highlight what are we trying to accomplish here. So basically, we are trying to translate in vitro the central nervous system. And all the models, the conventional models currently used, they try to capture the complexity of the human brain, but all of them have limitations. So some has the primary culture or the organotypic slices from rodent brains. They lack the human biology. The 2D cultures can be considered simplistic models, and maybe the 3D models are still technically challenging. But we agree that all the models have at some point some capacity to recapitulate certain functions and mechanisms. And at Encardia, we believe that combining the human-derived induced pluripotent stem cell technology, namely to be able to generate neurons and combining these human models with state-of-the-art in vitro platforms, such as the MEA that I will talk about later, we can increase the translational potential of the model. So, and in the end is what we are trying to achieve. So just to recapitulate what is a seizure and the seizure electrophysiology. So we have to keep in mind that is a phenomenon that happens in vivo, and for that we have a complete organ. So we have a brain, and a seizure is an expansion of a very synchronized pathological activity. So it's well defined, and then it travels to large cortical regions, and then it's followed by an abrupt self-termination. So in vivo and in patients, it's used both the intracranial electroencephalography and the EEG to identify those zones. So where is the onset of the seizure? And then they analyze the patterns of synchronicity. So as you can see in this map, where we can identify the local and the global bursts. But then in vitro, in our neuronal cultures, so first we don't have all these afferent connections that are present in the brain. But what we can try to do is to have different subtypes of neurons. and then they can represent some circuits. So in the end, it's something like we might not have a cortical forebrain or this region, but we might have some cholinergic neurons, and the cholinergic pathways will be active and be able to modulate our neurons, namely the GABAergic and the glutamatergic neurons, when we consider the cortical system. And so basically, we take these cultures, and they are electrically active, and then when we culture them in contact with the MEA electrodes, they generate a pattern of fire. So, these signals, so we can correlate these electrical signals with the pattern of firing, and when we get this fire pattern that is well organized and synchronous, we can establish, or we try to establish a parallel with what happens in vivo. So we call this organization in a network burst, and we believe that this is a robust marker for synchronicity, and it might bridge what happens in vitro and what happens in vivo. So maybe just to, because I believe that it's important. So I think it's important to define what is a network burst and what is the relevance for the in vitro and in vivo systems. So a network burst is a physiologically relevant pattern, so for normal brain activity, and it's regulated by a balance of excitation and inhibition. And I think this excitation and inhibition balance is a concept that I will come back later on when we will be describing our patterns of MEA activity for our cultures. And one thing that is very interesting is that the synchronized firing is observed in several models. So from in vivo to ex vivo cortical slices, so the organotypic cultures, the dissociated primary cultures, and also very interestingly, the cortical neurons that are derived from human-induced pluripotent stem cells. So basically, also in our models. And it's also interesting when we can make the link with these fundamental properties of the circuit function, so things like neurodevelopment, the plasticity, the learning, and the memory, and it's also connected to some neurological disorders. So when we have an impairment in these balance of excitation and inhibition and the dysregulation of these synchronized network bursts, then we have a disruption, and then we have these pathological phenotypes. So I will now talk just a little bit, or try to introduce the MEA technology without going in a lot of details. So basically, we use these MEA platforms, so the Microelectrode Array, to measure the electrical activity of our neurons. So we culture our cells on these plates. In this case, it's a 48-well MEA plate, and the cells will be on the top of a grid of electrodes. So in this case, for a 48-well plate, we have 16 electrodes, but there are other platforms like 96-well plates, and then we'll have 8 electrodes on the grid. And those electrodes, they will record the activity of our neurons. So one common and a very simple kind of readout is this heat map and the activity map. So for each well, we have those 16 electrodes, and then in red we have electrodes that are sensing the activity. And one of the best things about this technique is that it's a non-invasive technique. So we can monitor the network burst development over time. We can test experimental conditions. We can do compound screening. We can do multiple tests. We can do multiplexing without affecting the cells. So for long-time cultures, we can record over time our culture without disruption. Basically, just to revisit how we can translate our readouts on MEA in metrics that can qualify and quantify what is happening. So we have our neurons that they generate a voltage signal, and then we correlate this voltage signal with a pattern of firing. So it's represented in these raster plots. So this is an example of one well where we have our signal, and then in these associated raster plots we have all the 16 electrodes, and then we can identify the spikes, the bursts, and the network bursts. So these in the spikes we can correlate that we have a firing of a neuron, so it's just a simple firing. With the burst we have another level of organization of the firing properties, so we have the starting of having clusters of neurons firing together, and then the network burst is what we consider to be the culture will be synchronous, and then we have a pace firing in the culture. So it's what we believe is phenotype for a mature culture. So about the data, there are several steps in handling or processing the data, and it might be different across labs, and it also depends on the aim or the purpose. And it can really go from just using data analysis algorithms like the ones that are provided by Axion, like the neurometric tool, to others that are more sophisticated, like the spike sorting or the clustering. But in the end, it's with this data that we generate, or the metrics and the patterns that emerge, that we'll be able first to define these excitation and inhibition balance, and to try to understand better the intracellular signaling, and also correlate what we see in terms of firings and these kind of patterns to specific neuronal subtypes and specific targets. So when we talk about, or when we think about agonists and antagonists, this is what we'll be trying to read in our data. And so I will, and then we apply this technology to our neuronal models. So the first example that I will introduce today is our MSite CNS. So MSite CNS is a co-culture of human iPSC derived neurons and astrocytes. We believe that it's important to have a co-culture of neurons and astrocytes based on the support activities that astrocytes provide to neurons, and later I will revisit this topic again. And even if we don't have a full neuromodulation system and all the efferent pathways that we have in the brain in our cultures, I think it's important to recognize that we have the different neuronal populations like the GABAergic neurons, glutamatergic neurons, dopaminergic neurons, and it will be a better system to recapitulate the physiology of the brain. So now when we look at the firing properties of the culture, we can clearly see a progressive organization of the neuronal firing. So it goes in more complex and structured patterns. So the emergence of the network bursts at week 2 and week 3. So it goes from disorganized spikes and bursts to these more complex and organized structures. And it's based on these analysis of the culture. So when we quantify the active electrodes, the firing activity, the number of bursts, the number of network bursts, that we can define the assay window. So when I talk about the assay window, we have to try again to recapitulate what is happening at the molecular level. So when we try to define our time to have our compounds, we try to be in this kind of slope. So it's we try to work with in cultures that have a certain level of coordination, but they are not totally coordinated. So most of the compounds, they will disrupt this kind of balance. And when we talk about balance of excitation and inhibition, it's really this balance between the excitatory drive that we associate with the glutamate or acetylcholine or dopamine. So excitatory neurotransmitters, and then the inhibitory drive. So that is basically the GABAergic pathway. And if we want to go in more detail about what we see in this coordination is driven by this balance of excitation and inhibition, it also comes to the kind of the functional units. So we have glutamatergic and GABAergic neurons in the system, and we have some other types of neurons, cholinergic or dopaminergic, that will work as a kind of modulation in the system. So it's important for us, so bottom line, to establish these robust culture conditions and to support this balance of excitation and inhibition, because the compound, or our seizure model will be based on this. So in the end, so I will present some patterns of compounds that might disrupt this balance of excitation and inhibition. So in the end, what we try to do is to challenge the system for certain pathways or targets. So I will start with this muscarinic M1 receptor. It's a target for seizure, and pilocarpine. So it's an agonist for this receptor. Is a well-known seizogenic compound. So people use in their assays pilocarpine to develop seizure. To develop seizures even in rodent models, so both in vivo and in vitro. There is also another example for the cholinergic system with carbachol. So this one is an agonist for the muscarinic receptor, so it's not selected as pilocarpine. But now, if we go to the other side of the question, so if we have an inhibitory system in place, so the GABAergic pathway, and if we use antagonists for the GABA receptor, as in Picrotoxin and PTZ, and PTZ is also a well-known compound to induce seizures, we can also see this pattern of excitation. So if we compare our, so this central last plot, so the untreated, with all these patterns, we can clearly see that there is an increase in the number of network bursts, and also in the overall firing. And there is also another pattern that is different from the others when we use blockers for the potassium channel, so using 4-EP, so for aminopyridine, that is also a seizogenic compound, and 4-EP is also used to induce seizures in both in vivo and in vitro systems. So we can clearly see another pattern of excitation. And then in the other side, so if we now use antagonists for the glutamatergic system, so AMPA and NMDA receptors, we have the opposite. So we lose our structure, so we don't have almost no bursts anymore. We have a total obliteration of the network bursts. And it also happens for some drugs like the antiepileptic phenytoin. So it's also a pattern of inhibition. So with these examples, we are just trying to demonstrate that in this case for the M-site CNS, we have several pathways in place. It's not just the glutamatergic or the GABAergic. We might have modulation from other systems, and we can create these kind of patterns and correlate with our targets. So I will just bring these two examples again. So the one about the cholinergic pathway, so the using carbachol, so an agonist for the muscarinic receptors. And as I told you, and this is just in a very kind of overview, so I just selected the weighted mean firing and the number of networks has the metrics to qualify what we have in the system, but we have several metrics that we can use to describe these patterns. But the idea is just to show you that we can qualify this pattern of excitation, and also this suggests the presence of the active functional cholinergic receptors in our model. And then in the other hand, so using Picrotoxin, but now in this case as an antagonist for the GABA receptor, so we can also show that we have the GABAergic pathway in our system, so it's active, and we have this functional receptor. And I also use the same metrics. So for both the examples, we have an increased firing in the treatment condition, and also more network bursts. But if we selected other metrics, we can refine what is the pattern and what is the difference between an activation by the cholinergic system and inhibition of the GABAergic system. So then I will just move to our second model, so the end-site cortical neurons. So those neurons, they are human iPSC derived cortical neurons. They are essentially a glutamatergic culture of layer 5 cortical neurons. And then, as you can see by the activity map and the raster plot, those cultures are extremely active. And this is a kind of good news, and is what we will be expecting for a glutamatergic culture, but we might have to refine, and we have to do some assay development in order to preserve this kind of high activity that we also need to develop our compound testing. So the first thing that we did was the media optimization for cortical neurons. So one thing is to use the medium that we use to culture the cells and to provide the maturation, but the other is to be able to use the maturation in the process of electrophysiology. So what we did is we developed the end-site medium 2 that we also use on end-site CNS cells, and this enables the early detection of electrical activity and also keeps the high activity for a longer period of time. So in this case, at least 3 weeks. Another parameter that we identified, it was the co-culture with astrocytes. So we know that astrocytes support neurons, and in this case, we can clearly see that when we add astrocytes to our cultures, they enable the emergence of network bursts, and they can also extend the time in culture for at least 4 weeks. So now, to introduce just briefly our end-site astrocytes. So they are mature astrocytes. They express markers as GFAP, S100 beta, and acroporin 4. And we believe that our astrocytes are providing support to cortical neurons by reducing the glutamate concentrations in the medium. That we believe that in high levels can reduce excitotoxicity. So we can astrocytes can play other roles, like providing support with the neurotrophins and ATP, etc. But we believe that this might be the role that supports the most this culture. And so after sharing with you these examples of assay development on MEA and some patterns of signatures for seizure-related compounds, I would just like to talk about this application note that was done in a collaboration with the CSGO, where we tried to define the role of astrocytes on neuroinflammation. So basically, astrocytes are treated with a pro-inflammatory cytokine cocktail. So we use IL-beta-1, TNF-alpha, and interferon gamma. And then after that, astrocytes become activated and they release interleukins in the medium. And we can measure the levels of cytokines in these supernatants using the HDRF technology. So HDRF stands for homogeneous time-resolved fluorescence. So it's a no-wash technology, and basically combines the FRET technology with the time-resolved measurement of fluorescence. And based on what we collect from the supernatants, so we can measure the levels of IL-6, IL-8, CCL2, BDNF, we will define what the kind of activation we have in our astrocytes, if it's pro-inflammatory or not. And one of the best things about these kind of applications is that we can multiplex this further. So we use the supernatants, and then we can use also the lysates, and then we can further define the neuroinflammation and the activation level of our astrocytes. Finally, and I think this is my last slide. We believe that in our co-culture models, in our co-culture model of astrocytes and cortical neurons, we can explore this interplay between neurotoxicity induced by the excess of glutamate and neuroinflammation and the ways of modulating it. And this, we believe, that has a new level of understanding for this physiological role of neurons and astrocytes, and also it brings the opportunity to multiplex and combine results from completely different platforms, as the MEA and the HDRF. So I think with this slide I end my presentation, and I just would like to thank you for your time, and Stephen and I will be happy to answer your questions. Thank you.
Thank you very much. Yes, indeed. Just a reminder for the audience once again, in order to send in questions, you can send them in via the questions widget. But it looks like we've got some questions coming through already. So the first question for today's session is, can hiPSC be used for ASD?
Okay, so this question, I believe that is for me. So ASD is for autism, and I believe that it can be used to model this disease. So I believe that it's a complex disease, but there are a lot of genetic models that we can use both on MEA or other platforms. But yes, I believe so.
Thank you very much. The next question that we have is, does Nkardia develop manufacturing processes for cell therapy applications?
Yeah, okay, I can take that. Yeah, absolutely. So I would say that so we focus really on supporting early development up to preclinical. So the process for generating models is in a cell therapy project with us is slightly different than the workflow that I showed earlier for drug discovery cell models, for disease models. So in the planning phases of a cell therapy project, for instance, we might survey the IP landscape and try to really identify this clear pathway with regards to freedom to operate regarding protocols that are being developed. Well, as I said, we're involved earlier in early development stage, but we really focus on building a plan from the start that looks at where you want to be at the end. So although we're still developing, we're still keeping close tabs on things about how you could implement this protocol in a GMP setting in the future. So we look at how to close steps and how to reduce handling steps as much as possible. Even connecting with bioprocessing device manufacturers to kind of align that process as well. So if you have a partner in place already, that we make sure that we're developing protocols that can port to your process. There's also a bit more of an emphasis on things like tech transfer protocols and finding these QC assays around the product profile. and making sure that we have training and tech transfer to the client side or again, manufacturing partner. So yeah. So definitely. Yeah.
Thank you very much. We have another question coming up, and it's, might you be looking into expanding your product profile to include iPSC immune cells, such as those recently produced by ATCC?
So I guess I could take that. Immune cells, yeah, absolutely. So yeah, it's pretty amazing how much immune cells have taken center stage, especially looking at the therapies, cell therapies that are developing. As we know, the cell therapies had a foundation in autologous cell therapies, meaning coming directly from the patient and being modified ex vivo and then back into the patient. As you see, the challenges in logistics and transport, you start seeing more people looking towards iPSC solutions for allogeneic possible. So I think it's in an early stage, but you do see already big movements going. I think much faster than the industry maybe even had expected. And so, yeah, definitely we look in that direction. A lot of times those are questions around cell therapies. So in that case, we work with a slightly different framework. So in that way, it's more around co-development and partnerships.
Thank you very much. Another question we have is, without breaching confidentiality, can you say a few words about the types of company you have supported with stem cell-derived CNS cells?
Okay, so I think I can get that one. So I believe that we have most of our customers will be from the pharmaceutical industry. So everything that would be from compound testing, that could be to generate models, and also I think so thinking about customers that also this kind of partnership. So everything that would be to validate platforms, has the one that I was just describing with SysBio. So I think that it would be basically those. So the two major fields. One is really application to use our models to test several things. So compound disease modeling, etc. And then the other also, I think it's interesting, this angle with the technologies and to validate different platforms.
Thank you very much. Another question that we have is, how could you target the cholinergic neurons move towards the correct location?
Okay, so when I talked about the cholinergic neurons and the basal forebrain, it was just to give an example of what happens in vivo. So in vitro, we don't have that topography, we don't have that region. But with our protocols, when we develop these Insight CNS, we have other subtypes. So it's just not the glutamatergic and GABAergic neurons. So we also have cholinergic neurons. We have dopaminergic neurons. So we are not really looking at the migration in the cells or in the sense of development. But it's, if we have those cells in place, we can validate that the cholinergic pathway is active. So in order to have the cholinergic receptors active, means that we need acetylcholine in the system, and we need cholinergic neurons to release that acetylcholine. So it was in the sense that I was talking about that, to have subtypes of neurons supporting or trying to mimic the neuromodulatory pathway. So it's not really about migration.
Thank you. Another question coming up, and it says, thank you for the interesting talk. Are all NGITE cells generated and cultured completely serum-free?
Okay, so I think the NSITE CNS cells, the cortical neurons and sites, I believe that they are culturing in serum-free conditions. Yeah, that's true. So it's also our way at one point if we want to try to build up cell models that are more complex, and with the example that we gave with the cortical neurons and astrocytes, we might need to accommodate other cell types that are sensitive to serum. So we try to avoid these for one hand, and in the other to keep as much the xeno-free conditions.
Thank you. We have another one, which is, does Synchronized run an efficiency screen? Is there a maximum number of candidates that you can process any one time?
Okay, I guess I can take that one. So the question is about efficacy screen, maximum number of candidates that you can process at one time. Yeah, so this is a question we get often, and I am told by our discovery team that really the most time-consuming aspect in the development is really around the development of the screening assay and establishing and testing the automation protocols. And so that's really that's really the piece that requires the most effort. So that once those components are in place, and then the number of compounds in the screen is relatively irrelevant. It's more an impact on repetition and cost. In practice, we see up to 10,000 compounds, but we have a dedicated team for efficacy screening, and if you had a specific number you're looking at or a project, we're happy to discuss the situation with you.
Thank you very much. Another one. What are the implications of maturation and assay readouts for both MEA and neuroinflammation?
So I think maturation it's something that we have to acknowledge has a kind of a physiological process, or we try to mimic that in vitro. So we need to culture ourselves for a certain time to get a mature phenotype. And I think depending on the assay that we want to do, then we will need a certain maturation. So what I try to convey is so if we have a mature neuron, we believe that we have a very functional neuron. So it will be electrically active. It can be kept longer in culture. But then if we want to have, for instance, compounds that can influence the maturation status of the neuron or an astrocyte or other cells, we might need to use a different state, so a kind of in between, to be sure that we can modulate that. So I would say that there is an influence of the maturation in our assay readout. So it's basically the assay will be related to what we need to prove and what we will be reading on the assay.
Thank you very much. We'll cover one more question today, and that is, if I obtain CNS cells from Nkardia, what follow-up scientific or technical support will be available to help me optimize their use in my application?
Okay, I can take that one. So we develop our models in-house, as we said, and we use them routinely in our services. So we know how to handle the cells and we know how they behave. So you can expect handling guides and technical support if you have a specific question as you're using the cells. That said, I think it is challenging to simply, how do I say it, like basically wholesale integrate an off-the-shelf model into a process. That's from our experience. Or even trying to develop an assay based on these preset conditions of a standardized process or product in general. I think it can be challenging. So what we see is that clients who are using our cells in the context of a project, that really provides the most value for us. Being more proactive about the support than reactive. And that's where we try to be, because we're trying to really lower that barrier to implementing stem cell technologies directly into your process. So we try to make it easy, as I said before, really initiating a tailored approach. We try to be very simple about it, where we can modify, optimize things with you, like culture conditions, assay windows, and such. So standard support for off-the-shelf products, absolutely. And where we see the greatest value is when we understand a little bit about what you're looking for and what your process looks like. That's a general comment.
Thank you very much. Just a reminder for the audience, your questions will be available to be answered at a later date. So that just leaves me to thank Stephen and Isabel for what was a great presentation, and Gardia for sponsoring this session. To the attendees, when you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned and follow us on Twitter at BR Webinars for daily updates. Also, you can join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope that you have a very nice day.